Hello and welcome to the FunCoup Network Analysis Workshop. And I'm so happy to see that so many of you could take time from your busy schedule, both online and IRL, to join us, despite the fact that the topic today is actually not ChatGPT. And I mean, it is a little bit of our fault. We did have the word network in the title. And in case you wandered in and were expecting a ChatGPT talk, I can tell you that I did ask it about FunCoup, and I got an almost accurate and decent reply. I will get to the slight inaccuracy in a couple of slides. But what we're going to discuss today, let's have a look at the agenda. Well, first, as Eric mentioned, I will introduce FunCoup, and then you will see 3 live demos. You'll see a demo of novel interaction partners, MaxLink for disease genes, and comparative interactomics. And some will also include references to our pathway analysis tool, PathPix. And finally, we'll have a look at what can be done with FunCoup Network offline. Davide will present this. Our goal is that after this workshop, you will be comfortable using FunCoup for your research question. And we will also answer the question of why you should use FunCoup instead of other similar tools, such as String. Eric already mentioned some of the practicalities, but for those of you online, you can feel free to write your questions directly in the chat. We will monitor it, and we will take questions after each point on the agenda. So FunCoup for functional couplings or associations. And what do I mean by functional associations? Well, in our case, it refers to, it refers to the types of interactions we have in FunCoup and that we consider to be true or gold standard. And these gold standards are used to evaluate the quality of all included data. And gold standards are protein-protein interactions, both from direct and indirect interactions that come from validated, curated experiments. And it can be genes from the same metabolic pathway or signaling cascade. It could also be proteins from protein complexes or verified protein interactions and confirmed protein interactions, and also bacterial genes in the same transcriptional units or operons. And each gold standard can be used separately to produce individual networks, but they can also be combined, like in the default network that we have for FunCoup. So what data is evaluated and included in FunCoup? Well, one of the unique features of FunCoup is that it only contains experimental data. So there is, for instance, no text mining that could introduce noise or biases in the tool. And the interactions come from high throughput experiments of 10 different types of evidence. It's direct physical interactions of whole proteins or domains. It can be a correlated expression of mRNA or proteins. We have coevolution data, proteins having similar phylogenetic profiles. We also have genetic interactions, and we do have proteins with similar regulatory control, co-regulation via transcription factors or microRNA. Then we also have co-localization, so that is presence in the same parts of the cell, and correlation of protein abundance by mass spectrometry. And remember the slight inaccuracy of ChatGPT that I was talking about in the beginning. Well, it was talking about evidence sources instead of evidence types. For example, the evidence type protein interaction is pulled from the evidence source IREF index, and ChatGPT couldn't really handle the difference between those two. Every interaction in these experiments is evaluated using dimension gold standards to produce a confidence score for every link in the network. What does the distribution of confidence score look like for all our links? Well, here is the distribution of links for the Homo sapiens network in FunCoup and another popular network string. And as you can see, FunCoup provides links skewed towards higher confidence compared to string. And this was more formally assessed in the latest FunCoup publication, where besides string, another popular network was tested, HumanNet. You may have heard of it. And it is difficult to compare networks. We know this from before. So we used the already published method by Huang et al., where a gene set is mapped onto the network, and then genes are subsampled. And basically, you try to recover the missing genes using network propagation from these subsampled genes. So how did we do? Well, using all network links, both FunCoup and actually string outperformed HumanNet. But looking at just the top, the 10% of links with the highest confidence score, FunCoup actually outperforms both of the other tools. This was on our Homo sapiens network, which is the most widely used one, of course. But there are other networks in FunCoup. So these are the results for the top 10% of links. The other links in FunCoup, or the other networks in FunCoup, we currently have 22 species included, covering all domains of life. And besides Homo sapiens that I mentioned, we have the common model organisms, such as baker's yeast, fruit fly, C. elegans mouse, rats, Arabidopsis thaliana, zebrafish, and E. coli. We also have other model organisms, such as Dictyostelium discoideum, fish and yeast, C. intestinalis, and then other organisms which are common and popular in research. It's the malaria parasite, rice, cow, dog, rooster, and wild boar. And we also included another prokaryote, the grass bacillus or B. subtilis, and of course the two archaea, Metanococcus and Sulfolobus. And if you count carefully, or if you've been able to tag along so far, you see that I only have 21 species on this slide. And I will get back to the 22nd in a couple of slides. So all the species contribute with their data to other species via orthologous transfer of information. So that is via proteins that have similar genetic origins. So what kind of coverage of the genome and interactome do we obtain for these species? Well, it does vary, and is of course driven by the available experimental data. And all in all, there are about 60 billion links for all the species. And you can see here the distribution of confidence scores. We have high confidence for the Homo sapiens network, and 0.8 is kind of the cutoff which is used for tools operating on FunCoup as a default. You can change that. But genome coverage is also relatively high, about 90% for humans, but it does vary as well. And we have lowest for the archaeas. The two archaeas is between 21 and 25% due to lack of data, of course. So the 22nd species, as promised, it is, of course, the SARS-CoV-2 virus interactome. And we have actually mapped 22 out of 28 virus proteins and integrated them into the human interactome. Interestingly, many of the SARS-CoV-2 proteins are connected by two intermediate human genes. None share common first neighbor. And we actually trained our network using SARS-CoV-2 data, in contrast to other networks that just add the genes right on top of the network. And the downside of this is that you may miss important interactions. And for instance, we used raw data from one study where the spike protein was shown to be connected to only two genes that you see marked here, Golgi7 and the ZDHC5 gene. And after training the FunCoup network, we identified two additional interaction partners for the spike protein, the ACE2 gene and the BSG. And you may have heard this before, but the ACE2 was later shown to be a very important receptor for the virus entry into the host cell. And the BSG is also receptive for SARS-CoV-2, but it's in cell lines of epithelial origin. Another FunCoup feature is tissue-specific subnetworks. We have almost 242,000 genes in FunCoup with at least one tissue associated to them. And here's a view from the web resource, a subnetwork of 3 genes related to thyroid hypoplasia within the complete network. And here's what happens when we filter by tissue thyroid gland. And as you can see, the query genes are connected by other genes in the thyroid tissue, which is suggesting functional properties of this tissue-specific subnetwork. For Homo sapiens, we have tissue information for roughly 10,500 genes in 49 tissues. So next up is a feature still in its early development. It's directed links. And most of FunCoup links are undirected, only for Homo sapiens and for a small fraction of links, actually, where we do have regulatory information. And here is an example of such links. You can see them with arrowheads on the slide. Trying to break out. Yes, so finally, we have the associated network-based tools. And there are actually 2 already built in and another easily accessible. And the first up, it's the comparative interactomics tool, where one can visualize conserved evolutionary relationships for a query in different species. And there will be a demo on this later on, so I won't dive into it. And secondly, we have MaxLink, which is a network-based tool that relies on guilt by association to extend a set of genes, for instance, a set of disease genes, as you can see here. First, they're mapped onto the network, and then all direct neighbors are identified as candidates. And each candidate is then tested for enrichment in links to the initial gene set compared to the rest of the network. And those that pass a hypergeometric test are returned with a MaxLink score, and there will also be a short demo on this later on. Another tool that you will see mentioned is PathBix, and PathBix is actually a web server with a lot of interesting features, but it's a web server or network-based pathway analysis tool called Anubix, where instead of looking at gene overlap between a gene set and a pathway, Anubix looks at the crosstalk, so the links between them. And it measures the crosstalk between the gene set and the pathway, and then randomly samples a number of gene sets of the same size and degree, and measures the crosstalk to fit a beta binomial distribution and calculate an expected crosstalk in the network. And then finally, it compares the measured crosstalk with the expected one to get a sense of statistical significance of the observed crosstalk. Well, that was my brief introduction. So before I proceed to the summary, do we have any questions from the audience or online? We're still waiting. Okay, maybe the questions will come up during the demos. So as a summary, FunCoup is one of the largest databases for networks of functional association. It shows better benchmark performance compared to other popular networks. FunCoup has features such as tissue-specific subnetworks for biological context and link direction on a very tiny subset of links. And it does support comparative interactomics and network-based tooling, including disease gene detection by MaxLink, pathway analysis using PathBix, module analysis using Topaz, which David will tell you about later. And it can also, or has also been used for drug repurposing using Anubix. So why should you use FunCoup instead of networks like String and others? And you probably already kind of sense the answer, but there are a few points on this. So first of all, FunCoup contains only experimental data, so there is no text mining that could add noise or biases. It is more comprehensive at higher confidence scores, as you've seen that other larger networks, and it also has a higher benchmark performance. And because of its unique features, it's the tissue-specific subnetworks, directed links, although a very small subset yet, comparative interactomics, and MaxLink that are built in, and all the things that you could do with it offline.